An important concept in synthetic biology today is combinatorial design. Here, a set of related parts are explored in combination with other sets of parts to generate a new device. For this, we must first define families of parts. For example, let's say we want to express a phosphodiesterase in E. coli at a specific constitutive level. For this, we know we'll need a constitutive promoter, a ribosome binding site, an enzyme ORF, and a terminator to construct a gene that satisfies the rules of the central dogma. If we arbitrarily pick sequences for these parts, we are unlikely to get what we want. We might pick a phosphodiesterase that does not fold properly in E. coli. We might pick an RBS that forms a secondary structure with the chosen enzyme that prevents it from being translated. We might pick a terminator that similarly interferes with translation. We might pick a promoter that gives the wrong amount of transcript. Thus far, we have no working theory for avoiding bad decisions here, and thus we must try many combinations. We might start with a library of promoters that differ by transcription rate but are all functionally constitutive. We might choose multiple ribosome binding sites with different sequences. We might choose a diverse set of orthologs of the proteins and several options for the terminator. There is excellent cloning methodology, principally using Golden Gate methods, to combinatorially assemble parts arranged in bins like this. Thus, we can produce libraries of composite parts and screen through this diversity for the precise functionality we require. This still poses a challenge of how to define part families and how to choose the right combinations of parts to explore in an experiment. We've seen that we can cleanly distinguish parts as DNA, RNA, and CDS bins based on what level of the central dogma the functional molecule appears. However, these are far too general of abstractions to specify a combinatorial design. Thus, there are efforts to find other ways of specifying a more narrow set of parts. For example, we may wish to specify that a given slot in a combinatorial design be a promoter, and perhaps we more specifically wish to explore all sigma 70 promoters, or perhaps more specifically only those that are constitutive, and perhaps even just a narrow clade of specific commonly used sequences. It remains an unresolved question as to how best to organize information that will support design. Another unresolved distinction is that of a primitive element of genetic composition and primitive elements of biochemical function. Though different words have been applied to the two situations, I'll refer to them as standard biological part and feature, respectively. Standard biological parts refer specifically to methods of standardized assembly, and from a genetic perspective, this implies strict rules about the boundaries of genetic elements and means of expressing a larger sequence as a composition of smaller parts. However, the biochemical function of what is encoded in the DNA has nothing to do with how the DNA was fabricated or how some human being likes to think about sequences and their relationships. What matters chemically is the specific biomolecules encoded by the sequence, which is the feature concept. Thus, some problems of design require consideration of parts, while others require features.